So our subject for today is continuing on with proteins. And this picture here kind of shows what most proteins look like. In fact, I brought one with me. All right, looks like a big messed up Slinky. All right, now these proteins come in all kinds of different three-dimensional configurations. But basically, that's what a protein looks like. We learned that fundamentally a protein is made up of a coiled up chain of amino acids, several hundred amino acids long, that are coiled up in a precise order. You'd say, when did you tell us that? It was, let's specifically show you. It was page D4. On page D4, so on page D4, when we first introduced you to proteins, we said each protein was synthesized from 100 or more amino acids that are linked together in a precise arrangement, and then they become all coiled up. Now, last time we examined the structure of an amino acid, and we spoke about those. There are about 20 different types of amino acids. On page D5, we wrote on page D5 that the thousands of different types of proteins that are in our body or that are in the body of your dog or an elephant or a corn plant or an octopus, there are thousands of different kinds of proteins, and they differ from one another in the number of amino acids. Some proteins might be 50 or 60 or 100 amino acids long. Some might be several hundred amino acids long. Secondly, they differ in the types of amino acids. Some proteins contain all 20 different types of amino acids, and some proteins contain less than all 20, maybe only 8 of the 20 amino acids. Thirdly, the proteins differ in how those amino acids are ordered, their sequence. And fourthly, they differ in how these chains of amino acids are coiled up. Now, we wanted to give you some examples of proteins that are found in us and in some other organisms as well, but especially those in us. So, first off, what do we mean by level 1, primary structure, level 2, secondary structure, level 3, tertiary structure, and way down here at the bottom of the page, level 4, quaternary structure. So in fact, that's what we were showing you on page D13. So we'll take a look on page D13. And on page D13, what we mean by the primary structure of a protein is it's simply the chain of amino acids. Each of these little balls, this kind of looks like a pearl necklace, each of these little pearls or balls represents an amino acid. So we said a protein fundamentally is made up of 100, 200 amino acids joined together in a precise sequence. This is commonly known as the primary structure of a protein or a polypeptide chain. What does poly mean? Many. And peptide refers to amino acids. So many amino acids joined together is called a polypeptide chain. And in fact, the bonds that hold these amino acids together are known as peptide bonds. Now, in fact, there are no proteins in our body or in any other living thing that look like just a straight long chain of amino acids. We said all proteins have a coiled shape. The simplest coiled pattern there is a coiled pattern that looks like this. And that's known as a secondary structure. All right, so that's a simple coiled shape. In fact, that's the way my Slinky originally looked before I got it all messed up. Right? It looked like a simple Slinky structure. Now, let's give you some examples of proteins that have that kind of simple secondary structure coiled shape. Back on page D5. So back on page D5, we wrote that the primary structure is just the polypeptide chain. A secondary structure looks like, and I drew what looks like a Slinky. So an example of a protein that looks like that is keratin. Now, keratin is the name of the protein that makes up our nails. Your nails are pure keratin protein. It's also, keratin is the name of the protein that makes up your hair. Now, of course, your hair might have some color to it. Mine doesn't, but it might have color to it. Your nails don't. That's because, in addition, in your hair, not only is it made up of keratin, but it also contains some other pigments. But the stuff that actually makes up your hair is the exact same stuff, keratin, that makes up our nails. It's also what makes up the scales in fish and reptiles. It's all made up of a protein called keratin that basically looks like this. Now, there is another protein that also has this similar kind of coiled shape that's called myosin. The root myo is a Greek root that means muscle. And this is a protein found in muscle cells that is needed for muscle contraction. It's found in our muscle cells. It's found in all muscle cells. Another protein that has this kind of coiled, simple secondary coiled shape, like a slinky, is a protein called collagen. Collagen is a protein that makes up our tendons. It's in our tendons. It's also in our skin. Now, to help you better understand that a little bit better, let's return back to page D13. So here's page D13 again. D13, and on the right-hand side of D13, it shows a tendon. We're looking at the back side of somebody's leg, around their ankle area, and I think many of you know that the calf muscle, and the actual name of the calf muscle, you don't have to know, it's a terrible name called the gastrocnemius muscle, but we'll call it the calf muscle. The calf muscle is actually attached to your heel bone by a tendon called the Achilles tendon. It's called the Achilles tendon. It was named after Brad Pitt, who played the part of Achilles in the movie Troy. So I don't know if anybody remembers the story of the Trojan Wars. Where you've learned about this is in an English class you might have studied Homer. Homer was a Greek historian. Incidentally, we don't really believe what Homer said. Homer was blind, and he wrote about things that happened hundreds of years before he ever lived. But anyhow, Homer wrote two very famous books. One was called The Odyssey, which has the story, many stories, including Jason and the Argonauts, been made into a movie multiple times. And the other book was called The Iliad. And the Iliad is the story of the Greek-Trojan Wars. And it includes the very famous story about how some individual, a prince from Troy, kidnapped one of the princesses from Greece and took her back to Troy, which is in Asia Minor, not too far from Istanbul, today in Turkey. And basically that created this whole Greek-Trojan War. And anyhow, in that story, the Achilles tells the story of this heroic Greek figure, this military leader named Achilles. And you recall, if you saw the movie or read the book, The Iliad, so actually the whole story of Achilles was that his mother was pregnant, and she went to a Greek oracle. What were the Greek oracles? They were usually old ladies, where if you gave them a chicken, they would provide you with good advice. And she asked this old lady, this Greek oracle, if I give birth to a son, how can I make my son grow up to become a great hero? And she said, you will give birth to a son, and after he's born, hold him and immerse him in the magical waters of the river Styx, S-T-Y-X, and he will become invincible. So if you remember the story or the movie, so in fact, she gave birth to a son, she held him upside down by his ankles, immersed him into the magical waters of the river Styx, and he was invincible everywhere except at his ankles where the mother had been holding him. So Achilles is a great hero, and he fights all these battles, but eventually one day archers shoot arrows, and these arrows basically cut right here by the heel, and he falls and he's slain. And that becomes, so it was named the Achilles tendon, based on this story in the Iliad told by Homer. And today we use the expression, his Achilles heel, refers to somebody's weak spot, where they're vulnerable. That's the expression, his Achilles heel. So anyhow, what's that got to do with biology? I don't know. But anyhow, so this is the Achilles tendon. It's actually the longest tendon in the body. Tendons are these very tough structures that, as I wrote, attach muscles onto bones. And we said that this tendon, like all tendons, is mostly made up of a protein called collagen. And in fact, right here on your left, it shows the picture of collagen. You'll notice that each collagen protein kind of looks like a slinky, like this coiled structure. But what makes collagen very strong is because every 3 collagen proteins is braided together. And as you know, if you took 3 strings or strands and you braided those 3 together, those 3 cords into a rope, that makes it very strong when you braid them. So collagen is a very strong protein that actually makes up the tendons that attach the muscles of our body onto our bones. All right, so we've been giving you, back on page D5, we've been giving you 3 examples of proteins that all 3 have this kind of simple coiled shape, kind of like a slinky, that's known as the secondary structure, a simple coiled structure. Now, there is a more complex structure. There are, in fact, most proteins look not like a simple slinky, but they look like a big glob, like a big mess. This is known as a tertiary structure, or, because they look like a big glob, a globular structure. Now, your first thought is, I don't get this word tertiary. Well, it's Latin. You see, we have primary, which means first, secondary, right, here's primary structure, secondary, which means second, and tertiary means third. And way down here, we have quaternary, which means fourth level in complexity. This is similar to Spanish. In Spanish, we have primo, segundo, tercero, and cuatro, right? So it's similar to the Latin of primary, secondary, tertiary, like tercero, and quaternary, or quattro, the fourth. In its third level of complexity, it looks like a big, messed up slinky, and this is what most proteins look like. All the enzymes in our body are proteins that have this complex coiled shape, and enzymes are proteins that catalyze biochemical reactions. The word catalyze means they cause biochemical reactions to occur. There are thousands and thousands of biochemical reactions occurring in the cells of our body. Each biochemical reaction is catalyzed by a different enzyme, which are proteins. So literally, there are thousands of different enzymes in the cells of all living things. Also, there are protein hormones that have this globular shape. Now, last class meeting, we talked about steroid hormones. We had learned last class meeting, back on page D3 at the bottom, that hormones are chemicals that are secreted into our bloodstream by glands, and these hormones cause changes in our body. It was written at the bottom of D3. There are 2 categories of hormones. We said this last time. Steroid hormones, which are made from cholesterol, and the second category are protein hormones, which we're talking about right now. Last time we talked about testosterone and estrogen, corticosteroids, even calciferol or vitamin D. All of those were made from cholesterol. Now we're talking about hormones that are chemically proteins. They look like this, and they are made from amino acids. Now, we're going to give you a few examples of protein hormones, just as I had given you a few examples of steroid hormones last class meeting. So, the first one we've listed is insulin. Everybody's heard of insulin. What is it? Insulin is produced by our pancreas. Now we've looked at J5 before, so I'm not going to take time for us to look at that picture of the digestive system on J5, but that had a picture showing where the stomach is, and where the liver is, and where the gallbladder is, and where the pancreas is, and where the duodenum is. So the pancreas has a number of functions, and one of those is to secrete a hormone called insulin. What does insulin do? It is a protein hormone that lowers the amount of sugar, glucose sugar, in your bloodstream. So insulin actually lowers the amount of sugar, the amount of glucose in your bloodstream. Now there is a disease that you should know called diabetes. Diabetes is a disease associated with a deficiency of insulin. The word deficiency means not enough. A person doesn't have enough insulin. So let's think about what that means to the amount of sugar in somebody's bloodstream. What did we say insulin does to the amount of sugar in your blood? It lowers it. So if you don't have insulin, the sugar level, instead of going down, just keeps getting higher and higher. So in diabetes, the person develops increasingly higher and higher levels of glucose in their bloodstream. So you might first think, well, what's wrong with that? But we have pointed out that in living things, the level of everything should be the right amount. Any level of a chemical that is lower than normal or higher than normal is abnormal and basically is a disease or disorder. So insulin lowers the amount of sugar in our bloodstream. Okay, a second example of a protein hormone, besides insulin, made by our pancreas, is growth hormone. Growth hormone is a hormone, a protein hormone, looking kind of like this, made out of amino acids, that is produced and secreted by our pituitary gland. Does anybody have any idea where the pituitary gland is in the body? Yes, the pituitary gland is attached to the bottom of your brain. It's in your skull. It's attached to the base of the brain, right? And it's considered the most important gland in the body. It secretes about 10 different hormones. So growth hormone is just one of about 10 hormones that the pituitary gland secretes into the bloodstream. What does growth hormone do? Well, it does exactly what its name says. It promotes growth. It stimulates growth of your bones, causes you to grow taller. It also stimulates growth of muscles. You may have heard of athletes who inject not only anabolic steroids like testosterone, which we talked about last class meeting, but they might also inject growth hormone. That's what Barry Bonds was doing, because that increases muscles. Okay? So incidentally, all these different types of hormones are now regulated as controlled substances by the Department of Justice. But anyhow, so that's growth hormone. A third example of a protein hormone is oxytocin. And oxytocin, like growth hormone, is also released from the pituitary gland. Now it goes by a brand name, certainly not asking you to know this, goes by a brand name called Pitocin. Maybe you've heard of that. What does oxytocin or Pitocin do? We wrote that it causes labor contractions. It's what causes a woman to go into labor. So normally, when a woman is pregnant, around the 9th month of pregnancy, her pituitary gland starts to secrete this oxytocin hormone into her bloodstream that causes her to go into labor. And what is labor? Labor is the uterus contracting. That's what oxytocin causes to happen, is the uterus or womb to contract to push the baby out. Now, if a woman fails to go into labor, then what they would do is give her an injection of oxytocin or Pitocin, and within 5 minutes she will be in labor. That's called inducing labor. Now, those are just 3 examples of protein hormones. There are many, many more protein hormones than just those 3, just as there are more steroid hormones than the ones I talked about. Now, what we've listed next are plasma proteins. Plasma proteins, and it should read P-L-A-S-M-A protein, so if it's not spelled right, plasma proteins. These also have this so-called tertiary structure or globular structure. All of these enzymes, protein hormones, and now plasma proteins have this tertiary or globular structure. What are plasma proteins? Plasma proteins are proteins in our bloodstream. They are in our bloodstream. And I want to mention a couple of proteins that are in our bloodstream that have a globular or tertiary structure. The first is a protein called albumin. Albumin is a protein produced by your liver. And the function of albumin protein circulating in your bloodstream, produced by your liver and released into your bloodstream, is that it helps maintain your blood volume. If somebody were to injure themselves and be hemorrhaging or losing blood, one of the ways that they would treat them in the hospital is to give them albumin to help maintain their blood volume. Another protein that has this tertiary or globular structure are antibodies. Antibodies are also proteins. Now, antibodies are proteins produced by certain white blood cells known as lymphocyte, WBCs, lymphocyte white blood cells. So these lymphocyte white blood cells manufacture these proteins called antibodies. And what do antibodies do? Antibodies, we wrote, inactivate foreign agents. And so what does that mean? They inactivate or destroy anything that your body regards as foreign, that should not be in your body, including bacteria and viruses and so on. So this is how antibodies, these proteins produced by certain white blood cells, protect us against things that are foreign that enter our body. Antibodies are also referred to as gamma globulins or immunoglobulins. Anytime you have that ending, globulin, it refers to a globular protein, right? A tertiary or globular shaped protein. All right, well, so far we've been talking about proteins that look like this, that have a secondary structure. We gave you 3 examples, keratin, myosin, and collagen. We've been talking about proteins that look like a messed up slinky, that are said to have a tertiary or globular structure. That included all these enzymes, protein hormones including insulin, growth hormone, and oxytocin, and plasma proteins including albumin and antibodies. There is one more level of complexity, known as level 4, the 4th level, quattro, the quaternary structure. So what is the 4th level of complexity? To help us visualize this, let's look back on page D13. On page D13, so on D13, remember, we've talked about secondary structure proteins. We've talked about tertiary or globular proteins that look like a slinky. And now we want to talk about, at the bottom left, quaternary structure proteins. And an example of that is the protein hemoglobin. Now, the definition of a quaternary structure protein is that it's actually made up of 2 or more of these globular proteins twisted together. So I didn't bring another twisted up, messed up slinky like this, but if I had another slinky just like this one and I twisted the 2 together, we would call that a 4th level or quaternary level. In fact, in the case of hemoglobin specifically, hemoglobin is actually made up of 4 globular proteins, globular or twisted up slinky-like proteins twisted together. Now, since I mentioned hemoglobin, let me tell you a little bit about hemoglobin. Hemoglobin is a red-colored protein. It actually is red in color. And it is the protein found in our red blood cells, our RBCs. The function of hemoglobin protein in our red blood cells is that oxygen, O2, attaches to the hemoglobin that's inside of our red blood cells. So this is the way oxygen is transported or carried in our bloodstream. Oxygen is carried in our bloodstream, attached to hemoglobin, which is the protein inside of our red blood cells. And we don't just have one molecule of hemoglobin, we have hundreds of thousands of molecules of hemoglobin. Each one can carry an oxygen molecule. All right, now back on page D5. Back on D5. So we wrote on the bottom of D5 The quaternary structure is where we have proteins made up of two or more globular proteins intertwined, of which hemoglobin is an example. Now, hemoglobin altogether is about 600 amino acids big. I'm not asking you to know that. I promise not to ask you. But altogether, those four globular proteins twisted together make up about 600 amino acids. Why do I mention it? On the next page, D6, I want to explain what sickle cell anemia is. What is sickle cell anemia? Now, we know that inside normal red blood cell, this is a normal, good old normal red blood cell. So inside of a red blood cell is hemoglobin. And we said hemoglobin is about a total, each hemoglobin molecule is about 600 amino acids. And those amino acids are arranged in a specific, precise order or sequence. This shows part of that sequence of the 600 amino acids. You certainly don't have to remember it. But this is the sequence of amino acids. Last week we learned that most amino acids, you can recognize them by their name because they end in ene, I-N-E. Valine, histidine, leucine, threonine. They don't all end in ene. There are some exceptions. Glutamic acid, tryptophan. There are a few amino acids that don't end in ene. Now, what is sickle cell anemia? Sickle cell anemia is, we wrote, a genetic disease. It's a defect in the DNA, in the genes or DNA. And the result of sickle cell anemia is that the red blood cells are misshapen. They have this so-called sickle shape. They don't look round, they look abnormal, sickle-shaped. Why they don't look normal is because the hemoglobin in these sickle cell red blood cells is abnormal. The hemoglobin is abnormal. What's wrong with the hemoglobin? Well, out of the 600 amino acids that make up a normal hemoglobin, in people with sickle cell anemia, one amino acid in that hemoglobin protein is wrong. And you'll notice that in sickle cell anemia, instead of having glutamic acid, it uses a different amino acid, valine. This one mistake, this one defect of one amino acid in that entire hemoglobin protein molecule accounts for the entire disorder or genetic disease called sickle cell anemia. It means that the hemoglobin is defective. It does not transport or carry oxygen normally within the red blood cell, and it even causes the entire shape of the red blood cell to be misshapen, misformed, sickle cell shaped rather than the normal round shape. All right, now the next thing we're going to look at is denaturation of proteins. And we made a note to see page D13. What is denaturation? So on page D13, right in the very center, we know that a normal protein looks like a coiled up slinky. If the protein uncoils or unravels, we say that it has become denatured. That's called denaturation, to take away its natural coiled shape. And if it does become denatured, it stops working because the proteins only function or work when they're in that complex coiled up shape like this. Now, we might ask, well, fine, but what would cause a protein to unravel or uncoil? So I've listed 3 things that could cause a protein to unravel back on page D6. High temperatures, a change in pH, or the presence of certain heavy metals. Let's go through each of these. So the danger of hyperthermia or fever is that high temperatures will cause that 3-dimensional coiled up protein to unravel, uncoil, to become denatured and stop working. So when, for example, if a child or a pet is left in a warm car with the windows rolled up on a hot day, as the temperature in the car becomes hotter and hotter, the proteins start to unravel in all the cells of the body, including the brain cells. The child or the pet will start to have seizures, they will go into a coma, and they will die from all these proteins denaturing or uncoiling, unraveling. And that's also the danger of very high fevers. Another thing that would cause proteins to unravel, to become denatured and stop working, is a change in the pH. Now we know that the normal pH of the body is about 7.4. If the pH starts to become lower than that, that's called acidosis. Or if the pH becomes greater than 7.4, that's called alkalosis. And when this happens, acidosis or alkalosis, the proteins start unraveling, the individual will have seizures, go into a coma, and die. Let me give you one example of acidosis. So acidosis can happen whenever somebody's having difficulty in breathing. If you have difficulty in breathing, not only are you not getting enough oxygen into your body, you're also having difficulty getting rid of exhaling carbon dioxide out of your body. And whenever carbon dioxide starts to accumulate in the body, the CO2, the carbon dioxide, can combine with water in our body, and we certainly have plenty of water, and it forms carbonic acid. We actually talked about carbonic acid back in section C when we were learning about pH and carbonated drinks, and the fact that carbonated drinks are acidic because they contain carbonic acid. Anything with carbon dioxide bubbles, we know forms carbonic acid, and that gives it its taste. But anyhow, in this case, the buildup of carbon dioxide forms carbonic acid, which creates a state of acidosis, increased acidity in the body. Now, the third factor, letter C, that can cause proteins to unravel, to denature, and cause a person to go into a coma and die is exposure to certain heavy metals. Metals with a high atomic weight, such as lead or mercury. So if somebody were to either eat food containing lead or mercury, or inhale vapors, lead and mercury vapors, as the lead or mercury attaches to the proteins, it causes the proteins in the body to start unraveling, to denature, to stop functioning, and the person can go into a coma or die. All right, so what are nucleic acids? Nucleic acids are DNA and RNA. We'll see in a little bit. DNA actually stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Now, the nucleic acids contain 5 of the 6 most important elements making up the chemicals in living things. Anybody remember schnapps? Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. The 6 most important elements making up the majority of chemicals in living things. Well, the nucleic acids contain 5 of the 6. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Now, these nucleic acids, DNA and RNA, are actually made from thousands or even billions of nucleotides linked together in a precise arrangement. This definition of nucleic acids reminds us of the definition of proteins. We had said that proteins were synthesized from hundreds of amino acids linked together in a precise arrangement. Now we're learning that nucleic acids, DNA and RNA, are made up of lots and lots of nucleotides linked together in a precise arrangement. There's a reason for that parallel. Before we can tell you more about DNA and RNA, these nucleic acids, we first better explain what do we mean by nucleotides. Each nucleotide is itself made up of 3 things. Each nucleotide is itself made up of 3 things. It is made up of a nitrogenous base. That just means a chemical containing nitrogen, a 5-carbon sugar, and a phosphate group. So in fact, I drew an extraordinary picture of a nucleotide right here. And we're going to see better pictures in a moment. But here's a nucleotide. So in my picture of a nucleotide, how did I represent it? I drew a circle, and I labeled that the base, or nitrogenous base. That base, whatever that is attached to a 5-carbon sugar. Now, we have learned about sugars In fact, we needed to know about sugars for our first exam. We covered simple sugars, monosaccharides, on page D1. And we gave you two examples of sugars that were 5 carbon atoms long, ribose and deoxyribose. So in each nucleotide, there is a 5 carbon sugar, either ribose or deoxyribose. Not both, but either one or the other. And then attached to that 5 carbon sugar is a phosphate group. Phosphate, which we've learned before when we talked about phospholipids, is a phosphorus and 4 oxygens. This is called a nucleotide. It's made up of 3 things: a base, one of these 2 sugars, and a phosphate. And it might even have more than 1 phosphate. So then the question is, we understand phosphate. We've talked about these 2 sugars before. The question is, what's a base? There are actually 5 bases. They're labeled right here. They're identified right here. And on page D14, we were looking at an ATP molecule, adenosine triphosphate. We said that adenine and ribose together is called adenosine, and it could have 1 phosphate or 2 phosphates or even 3 phosphates, in which case it would be called adenosine monophosphate, adenosine diphosphate, or adenosine triphosphate with 3 phosphates. And in fact, that's what this is labeled as adenosine triphosphate. Now, this is a very special nucleotide. It's called a high-energy nucleotide because basically this is the gasoline that powers all living cells. If gasoline is what cars run on, ATP is what cells run on. That provides the energy. Now, how does this gasoline called ATP, how does it release its energy? If we represent the ATP molecule, the fact that it has 3 phosphates, this way, by writing an A for adenosine and 3 phosphates in a row representing the 3 phosphates, if this 3rd phosphate is split off the molecule, it releases energy. And in order for that 3rd phosphate to be cleaved or split off the molecule, it requires an enzyme called ATPase and a coenzyme calcium. Together, they cleave or split that 3rd phosphate, forming ADP, which stands for adenosine diphosphate, with 2 phosphates and phosphate. And that splitting of that 3rd phosphate off the molecule releases energy. Now we might ask, how do we form this gasoline? Where does this gasoline called ATP come from? Is it in our food? Actually, it's not in our food. The cells of our body have to make it, but they actually make it using foods that we've eaten, especially sugars. So the way that we make this gasoline called ATP is that an ADP molecule, adenosine with 2 phosphates, a 3rd phosphate is attached to it using energy to form adenosine triphosphate with the 3 phosphates. Where this energy comes from to form ATP is, as I wrote, it comes from the breakdown of foods like sugars, where we break sugars down with oxygen in a process called cellular respiration. So cellular respiration, which is a process we will be learning more about, is the process by which ATP is produced. This gasoline is formed in the cells using foods like sugars with oxygen. And then once we have this gasoline, it can be used to power all the different cellular processes. That's ATP. Now the next thing we want to look at, back on page D6, is to compare RNA and DNA. So on page D6, we wrote that RNA is made up of one chain of several hundred RNA nucleotides. The RNA nucleotides always contain the sugar ribose, and the RNA nucleotides use either A's, G's, C's, or U's, adenine, guanine, cytosine, or uracil. And we actually had a picture of that on page D14. So on D14, there was a picture of what it shows on the lower right, a portion of part of an RNA molecule made up of 1, 2, 3, 4 RNA nucleotides. In reality, it would be made up of hundreds of RNA nucleotides. Let's talk about DNA. All right, now DNA is more complex than RNA. Each DNA is made up of not one chain of nucleotides, but two complementary chains of DNA nucleotides. And it's not just several hundred nucleotides long, but millions of nucleotides, DNA nucleotides long. Now, in the DNA nucleotides, it always uses the sugar deoxyribose rather than the sugar ribose, which is used in RNA nucleotides. And the bases that make up DNA nucleotides are A's, G's, C's, or T's, adenine, guanine, cytosine, or thymine. And we also indicated that when we talked about how they are joined together, two chains are joined together complementary, what we mean is that if on one chain there's an A, it attaches to a T on the opposite chain. And if there's a G on one chain, it attaches to a C on the opposite chain. We can see this pictured on page D15. And on D15, so we see the ladder-like structure of a DNA molecule. Now, on the DNA molecule, so we see two complementary strands of DNA, and this is a DNA nucleotide right here. Right there is a DNA nucleotide. In this case, it's made up of adenine, A, but the sugar is not ribose, but deoxyribose sugar, and it's got a phosphate attached. Shown right below is a second DNA nucleotide with cytosine C, the sugar deoxyribose, not ribose, and there's the phosphate attached. And right below that, another adenine with a deoxyribose sugar and a phosphate attached. And below that, a G for guanine attached to a deoxyribose sugar and a phosphate attached. And below that, a thymine, T, attached to a deoxyribose sugar and a phosphate attached. Now that's showing one side, one chain of DNA nucleotides, and you'll notice that it is attached to a second chain of nucleotides, a complementary chain. Again, here each nucleotide, and you'll notice that they seem to be running upside down compared to this chain. They're running upside down this way rather than this way. And again, you can see a phosphate sugar, a deoxyribose sugar, a deoxyribose sugar, and thymine. Notice how the two chains are joined together. A pairs with T, and G pairs with C. And that's what we mean by complementary chains, how they complementarily join together. Now, at a lower view, to take in a longer view of the DNA molecule, you can see it really does kind of look like a ladder. The steps of the ladder are formed by where the two bases pair together. Whether it's A joining with T or G joining with C, that forms the steps of the ladder. And the side rails of the ladder are formed by the so-called backbone of the DNA molecule, which is formed by alternating phosphate and sugar, deoxyribose sugar molecules that form the backbone on both sides. Now, in fact, this ladder-like DNA molecule is then twisted. Imagine a flexible ladder that became twisted, and this shape is called a double helix shape. And it's basically like a twisted ladder. Let's see if we can explain now how this fits together into something that some of you have seen in lab. So on page D16, what we're now looking at are two chromatids. You'd say, whoa, what are chromatids? So we wrote right up here, a chromatid is another name for a DNA molecule. So what we see right here are two identical DNA molecules, or chromatids. Now, the DNA molecule can become coiled or uncoiled. And in this respect, DNA is very different from a protein. We've learned that proteins normally are coiled up, have a three-dimensional structure, and they have to have that coiled up shape in order to function or work. And in fact, anything that would make a protein uncoil or denature will result in that protein no longer functioning. But in the case of the DNA molecule, it is capable of both coiling up and uncoiling, and then coiling up again. And that's actually the way it's supposed to work. Now, the name that we give to when the DNA is uncoiled is we call the uncoiled DNA molecule, the uncoiled DNA chromatid or molecule or chromatid, we say that it has the chromatin shape. And the time in which the DNA has this uncoiled chromatin shape that's the way it appears in the nucleus of a cell as long as the cell is not dividing. Now, for those people studying lab, taking lab and learning about the life cycle of a cell, the period of time in the life cycle of a cell where it's not dividing is called interphase. So during interphase, when the cell is not dividing, the DNA has this double helix shape, the twisted ladder shape. However, when its cell is going to divide, it coils itself up. So let's see this double helix, this chromatin-shaped DNA molecule, coil itself up over and over and over and over and over again into this tightly coiled up shape. This tightly coiled up shape is called a chromosome shape. And the DNA molecule, or chromatid, only becomes coiled up, or takes on this so-called chromosome shape, during when a cell is going to divide. And for those people taking lab, you learn that we divide the whole process of cell division into 4 phases: prophase, metaphase, anaphase, and telophase. And in fact, one of the things that happens as a cell begins or prepares to divide is that this DNA molecule or chromatid becomes coiled up into this chromosome shape. And one of the last things to happen at the end of cell division, after a cell divides, as it finishes up cell division during telophase, is that the DNA molecule or chromatid uncoils and returns back to its chromatin shape. So let's see if we can summarize this again. The DNA molecule is also known as a chromatid. We use those terms interchangeably. It is the largest molecule that we know of because it's actually visible with a light microscope. And there's no other molecules that we could see with a light microscope. We can see some others with an electron microscope, and there's other molecules that are smaller than that that can't even be seen with an electron microscope. Anyhow, DNA is huge. And the DNA molecule can either become coiled up, which it does when a cell is dividing, or it can uncoil back into where it just looks like a double helix shape. When the DNA molecule is uncoiled, it's said to have a chromatin shape. And when the DNA becomes coiled up, which it does during cell division, it's said to have a chromosome shape, or for short, people will just call it a chromosome, meaning that it's in the coiled up shape.